Welcome to everybody to another of our inaugural lectures series for this year. It's always a great pleasure to host these and a real chance for us to celebrate our professors. So I'm Laura Green, I'm head of the College of Life and Environmental Sciences. Welcome to those of you who are here in person and also welcome to those on Zoom. And today we are celebrating the appointment of Nigel Maxted to Professor in the School of Life Sciences in 2020. So I've got a little introduction that Nigel has written and I'll share with you, and then we'll move over to the main event. So Nigel was born in Cornwall and grew up in Kent. From an early age, he developed a love of nature and being in nature. When he started to think about work, the only real constraint that he had was that he wanted to continue living and working in the country. Nigel left school with minimal qualifications and started working on farms, initially in Kent and Norfolk, and gained an OND in agriculture at Hadlow College, Kent. He later moved to Canada and then the US, where he worked as a cowboy with a recalcitrant horse called Toby and minimal lassoing skills. You didn't know that, did you? I bet. Having emigrated to the US, he was forced to return to the UK to have an operation on his right leg. But this was not covered by the US Health Services. And so he arrived back in the UK. The operation was vital in the US, but turned out not to be essential in the UK, so it was never done. However, while awaiting the operation, Nigel took GCE A-levels in botany and zoology and gained entry to study biological sciences at Wolverhampton Polytechnic. He then did an MPhil research assistantship at the University of Southampton, working on the tropical legume wing bean on breeding use crop wild relatives. Sorry, but I'll say that again, on breeding using crop wild relatives. Following completion of the research assistantship, he took a PhD, also at the University of Southampton, on the temperate legume vetch and graduated from that in 1990. During both the MPhil and PhD degrees, he took time out to do consultancy work for the Food and Agriculture Organization of the UN and the International Board for Plant Genetic Resources on various aspects of genetic conservation. In 1991, Nigel started a lectureship in conservation and use of plant genetic resources at the University of Birmingham and ran the internationally renowned Masters in Conservation and Use of Plant Genetic Resources, until changes in the international scholarship sponsorship caused this course to close in 2008. Nigel was appointed senior lecturer in 2021 and Professor of Plant Genetic Conservation in 2020. His professional expertise is in plant genetic conservation, in situ and ex situ, and he has published over 350 peer-reviewed scientific papers, 220 edited book chapters, and 25 books. Most recently, his focus has been on crop wild relative and landrace conservation and conservation policy development. Nigel has led several national, European and international research projects and regularly undertakes consultancy work for international agencies such as the UNFAO, GEF, the World Bank and European Environment Agencies. Nigel has had many external roles and is currently International Scientific Advisor for the Alliance of Biodiversity International and CIAT, co-chair of the IUCN SSC Crop Wild Relative, very bad page turning, specialist group, chair of ECPGR Wild Species Conservation in Genetic Reserve Working Group, and chair of the DEFRA UK Plant Genetic Resources Group. I think you'll say that is a pretty long list of esteem. Nigel, it's over to you, and we look forward to hearing your story in full.
Thank you. Right, so you'll not be surprised, those who know me see the first thing that I'm going to do is take my jacket off. Okay, so I guess if you're going to give an inaugural, you have two choices. You can sit here and boast about the achievements of your life, or you can look forward. And I want to really look forward because I still am very active and there's a lot I still want to achieve with my research. So the title of this talk is on doubling genetic diversity for crop improvement. So those of you, not all of you know about how crop improvement works, but basically to keep on generating new cultivars, you need access to broader, new genetic diversity. So you need to have continual flow of genetic diversity into the breeders. And currently, because of climate change, this is being slowed up. We can't find the right genetic diversity. And we do tend to need a greater breadth of genetic diversity than ever we have done previously. So the idea of being able to double the amount of genetic diversity available to breeders, as I'm proposing, is really something that's very fundamental to sustaining crop breeding, development of new cultivars, but also as a relation to that, food security. And then we're talking about global food security. And as you'll hear in a minute, it's based on benefit to a very large number of people. Okay, so what I'm going to talk about is this challenge to plant breeders, human population levels, and climate change, and then the policy context of what I'm doing or the work I work in, and then basically the focus on ex situ conservation, conservation in gene banks, and then how to maximize the diversity that's available for use. And for that, I'm going to argue that we need to focus on crop wild relatives, and how to actually double that diversity is, instead of ignoring in situ conservation, to do in situ conservation as well as ex situ. So that's what I'm going to argue. So I usually start off with this talk to remind everyone about the basic problem we have. We have too many human beings. So 7.94 billion human beings. Yesterday, I think, or the other day, I looked it up. And predicted to be 9.8 in 2050. That recently the figure has been upgraded. It was 9.6 for many years, and now they're putting the figure up. So we're not leveling off in our estimates. The figures are increasing in terms of human population rise. Associated with those figures, we have between 720 to 811 million people currently facing hunger in 2020. These figures, I couldn't get today's figures for that, but the figures in 2020, 720 million to 811 million. And those people that are suffering moderate to severe food insecurity, 2.37 billion human people on the planet. So this really is fundamental of importance to tackle this issue and resolve it. At the same time as having all these people, obviously they have to eat, they have to be fed. And FAO have estimated, Food and Agriculture Organization of the UN have estimated, to feed the people that will be on the planet in 2050 will require an increase, a doubling, 100% increase in the amount of food availability. And this is in developing countries. So 60% globally, for countries like the UK, we're not going to need any more or significantly more food in the UK because our population is not increasing. But if you come from Nigeria, for instance, then there is a significant increase in population there, and they're going to need significantly more. So 100% doubling of the amount of food available in developing countries. While at the same time, we know that climate change is going to reduce the amount of food we can produce by 2% per decade. So you see the kind of crisis we're heading towards and why it's so important. So I would personally argue that the planet is beyond its carrying capacity for human beings. We have too many humans on the planet. And if the religions would do something about monitoring or controlling addressing population growth, then we would be in a much better state and wouldn't be destroying the planet at the rate we're doing currently. So this is recognized in the policy context. It's picked up by the CBD strategic plan, which talk about the need for increased genetic diversity, including from crop wild relatives for maintaining food production. It's also picked up in the global strategy for plant conservation. 70% of genetic diversity of crops, they say, is conserved. I doubt that figure is correct, but I've included it because they also talk about the need to look also at crop wild relatives in terms of diversity. And then the UN Sustainable Development Goals, as you know, 17 or 18 goals, of which the one that focuses on hunger is 2, and 2.5 clearly calls again for increased levels of genetic diversity being made available and crop wild relatives to be actively conserved. So the challenge of breeders is to get access to broader ranges, the full breadth of genetic diversity that possibly is available to help to conserve human beings, to maintain human survival into the next century. So what are genetic resources? So for those of you not familiar, genetic resources, genetic diversity is essentially the material, the alleles, the genes that are available and give value to agriculture because we use them in some form or other. This diversity enables the crops to be able to adapt to new diseases, new pests, ensures stability of production, and reduces the risk of climate change having devastating effects. So effectively, what we want to do, if we want to maximize our chance of feeding that population, is to increase the number of crops that are cultivated and also the crop varieties which are available in each crop itself. So the standard way this is done at the moment is we're looking, as I said, for a broad range of genetic diversity, alleles, gene combinations. For orthodox seeded species, seeds are collected, they're sampled, they're then dried, they're cleaned, dried, and frozen at -20 or cryopreservation -196. And they're taken out and characterized. We look for the traits which are present in them, either using for morphological traits or looking at genomic traits to identify the alleles present. And we're promoting that use. So basically the job of the gene bank is to maximize the genetic diversity which is conserved, characterized, because if we're characterizing it, we know which alleles are present, so the breeder can actually ask us for a population that has a particular allele, and we can supply that population and document that, making the documented information available to breeders. They can choose the material they want and use it in their breeding programs. So we're promoting use. There is this job of conserving, which is always linked to some form of utilization. And if we look globally at the ex situ gene banks, there are about 7.4 million accessions in gene banks in about 1,750 gene banks worldwide. The largest gene bank in the world you've probably heard about is Svalbard, the Global Seed Vault, which is a global backup of this genetic diversity, which again emphasizes how important it is that this material is available for our future, the future of humankind. Okay, so then we move on to the next section, and I want to talk about the challenges facing breeders. So a lot of breeders are reporting that they don't have access to sufficient genetic diversity. In other words, they want to breed new varieties. They realize there's a need for new varieties, but they don't have the genetic diversity available to actually use in their breeding programs to produce that. So that's the first point. Not enough genetic diversity is getting through to the breeders. The second is that genetic diversity is located in plant genetic resources, primarily crop wild relatives and land races. And crop wild relatives, we know, are suffering erosion, extinction. So depending on which study you look at, between 16 and 35% of different groups of crop wild relatives are threatened with extinction. 99% of crop wild relatives are conserved just using the one strategy, ex situ conservation, and that's primarily in gene banks. And if you look at gene bank holdings for crop wild relatives, about a third of the species of crop wild relatives have no material at all in the gene bank. Now, if there's no material in the gene bank, it's impossible to use them, yes? So we need to be much better at conserving that material. About a third are very poorly conserved. In other words, they don't contain the whole genetic diversity which is found in the species. It's just a partial view of genetic diversity within that species. And as a result of that, if you put all that together, then we come up with this figure of 95% of crop wild relative genetic diversity needs further collecting to actually represent the diversity that should be available to breeders. You see on those figures, I've quoted figures entirely for crop wild relatives. Why? Because the other source of genetic diversity are land races, and we have no idea at all how many land races exist, where they are, how threatened they are. We have no data on that at all. We know about crop wild relatives simply because we're focusing at the species level, and that species level data is made available by botanists from Kew going out and doing their normal botanical studies. So there's a huge bonus to us that crop wild relatives are wild plants because they're studied by ordinary botanists, and we can use their data. So the point I'm making is that crop wild relatives are very highly threatened. My feeling is with land races, they're even more threatened than crop wild relatives. But we can't prove that because we don't have the data to even assess it. Okay, so my argument would be, I've tried to make the case that we need this diversity. Currently, it's only conserved at all using ex situ techniques. And so we should start looking at in situ techniques to complement what we're doing ex situ. And in fact, the very first paper I wrote with Brian Ford Lloyd when I came to Birmingham was a letter to Nature, and it was basically explaining the need for complementary conservation. And that means complementary conservation employing both ex situ and in situ techniques. But frankly, in situ is almost completely ignored today. If you ask me how many protected areas in the world are crop wild relatives actively conserved in them, or land races, a handful, less than 5 globally. Why? You know, this is ridiculous. And if we have other kinds of conservation, the IUCN have got this new category of other effective area-based conservation measures, which means basically doing conservation outside of formal protected areas, but on roadsides, around the edges of orchards, etc. Again, I don't think there's a single example I can give you where they're being actively conserved in that way. Now, what the gene banks are actually focusing on in terms of their conservation is improving accessibility to the material held in the gene bank. This is good. I mean, it's good that they're doing that, but that has a limit, because you can only improve the amount of data that's available on a subset of accessions that have been collected and put in the gene bank, and we already know they're a very poor sample. Yes? So we can get much greater diversity by conserving in situ as well as in situ. So my focus on the rest of the paper is arguing, one, that we should be looking at crop wild relatives. Why? Because they maximize the amount of diversity that's contained per unit species. And secondly, that we should be doing active in situ conservation. And if you look at these two graphics on the side, this is just to show that there are a whole series of techniques, about eight or nine different conservation techniques that are applied. Some are in situ, some are ex situ, but depending on whether you're working on crop wild relatives or land races, you have alternate combinations of those that are available. So it's not a simple job to work out which conservation technique is most appropriate. Okay, there's a classic paper from Tanksley and McCouch, published in 1997. And basically, they were the first people to point out that in terms of genetic diversity, maximum genetic diversity is found in the wild species, as is indicated here by different color alleles in the gene pool of any wild species. When it goes through the domestication bottleneck, you're not domesticating the whole species. What you're domesticating is just a few populations. So you're domesticating a subset of the genetic material to start with. So immediately you're losing a huge percentage of genetic diversity. So you see this shrinking of the gene pool in land races, crop land races, and then a further shrinking in modern varieties, because farmers want to harvest all their material in one day. So they don't want any variation at all. So you have this gradient of diversity from wild species, land races, modern varieties. And the quote that I only found recently by going back and rereading this original paper by Tanksey and McCooch is that they give an example of tomato, saying that for tomato, 95% of the genetic diversity in the gene pool of tomato is found in the wild species of Lycopersicum and Solanum, which are related to tomato, not in the crop itself. So that itself is an interesting statement showing how important it is that we actively conserve these wild species. So one of the crop wild relatives, they're the ancestors of the crops that the crops were domesticated from. They are potential gene donors still today. They haven't suffered the domestication bottleneck, so they still have all that original array of diversity. And in very broad terms, a crop wild relative is a species, a taxon, that's in the same genus as a crop. So if we say that Solanum tuberosum is potato, any other Solanum species is a crop wild relative of potato. Yeah? We can come up with a more precise definition and hone it down to a much narrower number of species, which are really important. But I mean, that doesn't make, I won't go into detail on that, doesn't make any sense. But just to show the Tanksey and McCooch idea again, in this bar chart here, you can see the amount of genetic diversity in the wild species, in the land races, in the cultivars, exactly showing the relationship that was put up by Tanksey and McCooch. Okay, so my argument is, as well as looking at crop wild relatives, we should be looking at in situ conservation in a complementary manner. So we shouldn't be putting all our conservation eggs in one basket. We shouldn't be just focusing on ex situ. We should be doing ex situ and in situ. And why I've shown this is one of the problems of picking sites for in situ conservation. So this is a very nice fishing village in Turkey, where Sheila and I have spent many lovely hours drinking in the bars and eating fish. Associated with that is this small island off the coast. And on that island, there are 20, 30 species of crop wild relative, important crop wild relative related to the cereals. Now that's it's sea level. With climate change, if sea levels rise, that whole island is going to be lost. And yet that is one of the islands that's being recommended for conservation of in situ. So you have to be careful where you're doing your in situ conservation to make sure it's going to be long lasting. Okay, so the goal, what is our goal? Create a permanent system of in situ conservation of plant genetic resources, so land races and crop wild relatives, to complement what is already being done ex situ in gene banks, and to link through to use. Use is the ultimate aim. We need to conserve that material so it can be used in breeding, and then that then has the benefit to society. So the network itself will be composed of a whole series of sites, localities, populations. They would be managed to an agreed standard, and we produce that standard. There's a written standard on how the populations will be managed to make sure genetic diversity is maintained. The people who actually manage the sites, the custodians of those sites, there's a link to the gene banks, the genetic resource centers, because of the chance that in situ conservation is going to change in situ sites, you must always have a backup in the gene bank as well. So you need to have that ongoing relationship with gene banks, and then other stakeholders, maybe plant breeding companies, other organizations that are involved. So the network itself will be composed of plant populations, organizations, other networks, plant breeding networks, farmer networks, and individuals involved in joining and establish the network. So why do this now? Because there is now this current need to meet the breeders' needs. We know the resources, crop wild relatives and land races are threatened. We can identify the conservation gaps. We know exactly where we need to go collecting to do more conservation. We know where to go in terms of in situ conservation. We're able to identify those gaps. We want to have an interlocking system that ties together local conservation with national conservation, with European or continental conservation, with global conservation, because the same techniques are used at all levels. And we have to have legislation back up to make sure these sites, once they're organized, are there permanently. And by doing so, we can meet the obligation on the, as I said, on the UN Development Goals. So conservation and use linked to food security. Okay, so I wanted to go through perhaps I haven't done science. I'm a professor, I should talk about science. So I haven't talked about science. It's been largely, I guess, trying to berate you about the need for good policy intervention. But here's some science, some papers that we did recently. This is all work done on a global priority set of crop wild relatives. So we worked out what are the most important crop wild relatives related to the most important crops globally. And we came up with this list of 1,394 plants. And then we looked at the data on where they're found, and we worked out where the hotspots are. Now, not surprisingly, the Mediterranean, well, perhaps it is surprising. It comes out, it's just a strong hotspot. But these things are obviously found around the Mediterranean. It's the hottest hotspot globally. But we have sites in North America, South America, and China. And I would also add the Chinese data was very spare. The Chinese seemed not to be keen to share their data with us when we were doing this analysis. And so we did not have a represented data set from China. So in other words, I think China is also important, but doesn't come out in this analysis as strong as it did. So this is a species or taxon richness map based on the crop wild relatives that we can get detailed data for. So when I say detailed data, I mean more than 25 location occurrences for each taxon. So this is looking at areas, gaps in crop wild relative ex situ conservation. So these are places where they're under collected and we need to go to do more collecting. So here we see France and the Iberian Peninsula come out as globally sites which are under collected. We're completely baffling. But it's true that the answers are there. And then if we look at the South America, for instance, you have this site near Brasilia that's very high. That's because of arachis. Arachis peanut comes from that area, and this is a priority area for conserving the 16 wild arachis species. But again, the Mediterranean and then into Indonesia, also areas that are important for ex situ conservation. We can look at the areas important for in situ conservation. So this is analysis looking at the 170 spots globally. Some of them are protected areas and some of these sites outside of protected areas. So the 170 top sites where we should be focusing our in situ conservation of crop wild relatives based on the same data set that was used for the original taxon richness. And if, for instance, for each one of those taxa, we don't just have one occurrence in the network, we have to have 5 occurrences for each taxon. All of those 5 are not chosen randomly as 5. We're choosing them deliberately for the ones that have the maximum genetic diversity. And we're looking at each site and predicting it'll be least impacted by climate change. So these are the populations that will be least impacted by climate change, and they're genetically diverse, the most 150 sites globally. And then this enabled us to look again at the, historically when people talked about plant genetic resources, a lot of the original work was done by N.I. Vavilov, a Russian scientist who died tragically he crossed Stalin. If we have time, there's wonderful stories about Vavilov. But anyway, unfortunately, he fell out with Stalin and was put in prison and literally starved to death, poor bugger. But he published a paper talking about the hotspots of genetic diversity of crops in 1926. Now, it was only in 1958 that Myers published the first biodiversity hotspots. So the work of Vavilov was several decades ahead of Mars's work. Now, when Vavilov was doing this work, 1926 in Russia, he didn't have ideal access to data sets. So with the additional data sets we now have, we could revise, and we've added additional hotspots in South America, either side of North America, in West and Eastern Africa, and in Northern Australia. And then lastly, the last analysis is looking at the correlation between biodiversity hotspots, so hotspots for elephants, tigers, lions, etc., and some plant species, and crop wild relatives. And there isn't a perfect correlation. So if you were to say, okay, well, we'll leave all this crop wild relative conservation to biodiversity hotspots, it would be a mistake, because you're not covering that. Those are side by side. There's not a complete overlay between the two. Okay, so back to talking about what we want in the network. So is it preferable to have individual standalone sites or have a network either at the UK, European, global scale? Well, we think it's better to have networks. Why? Because we can coordinate them systematically. We can have stronger partnerships. We can deliberately have interaction between the different geographic levels. We can facilitate access and benefit sharing. So if we have one of the big limitations at the moment for individual farmers in being involved in a system like this is they don't know how to apply the international legislation, which comes out of the Nagoya Protocol or the international treaty. So if they join the network, we can negotiate these for them. So they will have maximum benefit from access and benefit sharing. So if someone actually uses a gene and makes a lot of money out of that gene, some of that benefit will flow back to the original farmer who provided it. So this is where we're talking about actually doubling the resource, is making this additional material available. And then there are lots of benefits for the individual maintainers. I won't go through all of them, but just to show we put a strong argument, I hope, that makes people want to join the network, and you'll see they do later on. And then if we're looking at actually protected areas, conservation in existing protected areas, so you might say, okay, well, why do we need to have any relationship with biodiversity conservation areas if there's no correlation or little correlation with crop wild relatives? Because they're already set up. They have an ethos of conservation. So to actually get the money to set up a completely separate network is not going to happen. So the best way of doing it is to work with protected areas and conserve as much as we can within them, because they have this long-term conservation ethos. They're not likely to have any substantial management changes of the site, so we'll maintain the populations. We can easily amend the management plan for the site to manage those populations. We can avoid the prohibitive cost of setting up completely new sites. And we know there are some crop wild relatives found in existing protected areas. And then in other cases, we can conserve the ones found outside of these protected areas, as I've said here, with these less formal sites. But the less formal sites still have to have legal protection, otherwise they won't last. So we want to complement the conservation of crop wild relatives in protected areas with conservation in these OECMs. And then the global site. So the next question is, where in the world is the highest concentration of crop wild relative diversity? It's here. This is Crac de Chevalier in near Homs in Syria. I know that the rebels were in the castle and it was attacked by Syrian fighter jets. So presumably it doesn't look quite so nice now as it does in this picture. But actually below this, the fighting won't have disturbed any of the populations of crop wild relatives. And around this, below this site, is the place in the world that has the highest concentration of crop wild relative diversity. Okay, so I talked about this interrelated geographic levels. So this is just a simple graphic showing, if you have it, the bottom layer here, a whole load of individual populations of crop wild relatives or landrace populations. Some of them will form part of the national network. So you identify, right, this particular population is important. We're going to give it priority and we'll make it part of the national network. The national network will have some sites that go into the European network, and the European network will flow into the global network. So you do have this integrated network structure. And then actually choosing sites can be done scientifically. There is a whole process. I haven't got time to go through it in detail, but you can collate the passport data, the data on locations. You can work out where the locations are concentrated in terms of numbers of populations, but also number of taxa, and then focus your activities. And it can be done either in a top-down version. So if you're working in Europe, you could collect the data for Europe and do a European top-down analysis, or you could get the national programs of each country, say, right, we know we have this particular site, which is good, and we want it to join the network. So we have both a bottom-up and a top-down approach, which politicians like to hear about. Then this is one of the things that's taken most time, I think, to resolve. And so I'm going to spend a little time explaining this diagram. So here on the left-hand side, you have the in situ protected area, say, where you're doing conservation of your crop bud relatives. And as I said to you, if you want to actually have effective conservation, you have to have this link between conservation and use. So how do you do that? So the obvious first way of doing it is, okay, the user, the plant breeder, would go to the protected area manager and say, right, can I have 100 seeds of this particular crop bud relative? Can I take it away and use it in my breeding program? It will never work, because the number of conversations we've had with protected area managers, they don't want to exploit their material. It's not in their ethos. And they don't see the benefit to them in being involved in that kind of structure, and they don't know about the legislation. There are two important international legislation associated with the Nagoya Protocol and the International Treaty for Plant Genetic Resources, both of which have implications for them globally to provide the material, but also for them to have access and benefit sharing. So if anyone exploits that material, makes money out of it, they can get some resource back. So how do we do it? If they can't go straight to the landowner, to the farmer, to the protected area manager to get the resource, how do they do it? Right, so we have 4 different options here. So in other words, you could make everything that's found in situ duplicated ex situ. Everything is duplicated. So everything is sampled, and that's your option 1, and goes in and is processed in the normal way and goes into the gene bank as an additional ex situ collection. The trouble with that is it's enormously expensive, because you're processing a whole load of material and you don't know if it's ever going to be used. So the second option is, okay, we'll give the material to the gene bank, but we tell them not to release it to anyone. So they just hold it in what's called black box conservation. So the material goes into the gene bank, is processed, but it's only available to the people who actually gave you the material in the first place. So it's not actually used. The third option is demand and supply. So if you have a plant breeding company and they see a population in the wild and they want to have a sample of it, then they demand it. They ask the protected area manager or the gene bank to provide that material. And the gene bank will go and collect it, they bring it back, they process it in the normal way and then supply it. Now, the problem with that is that you can only do it certain times a year, because you want seed, and the seed is only going to be present for 2 or 3 weeks of the year if it's an annual species, and maybe less if it's a perennial species. So there are problems with that approach as well. It's very time consuming. So I think the best approach is this 4th one here, and it's the one we're now going for, is you have the sample, which is the normal backup sample from the ex situ sample from the in situ resource, and you don't go through the whole process. You break down the different steps involved in processing, and one of them is regeneration, and regeneration is the one that's enormously expensive. So this is, why do you need regeneration? So you put the seed into a gene bank, and it can't stay there forever. It has to be taken out occasionally, and the seed regenerates so the viability remains high. Yes. So if you go through this process, it's very expensive, and also you get a strong selection pressure for where the material is regenerated, which is usually in temperate countries and it's been collected in the tropics. So you can see there are problems associated with that. Now, if we're dealing with in situ material, we don't need to regenerate because we can just go back to the in situ sample and in situ site and take a new sample. So that way we get round the regeneration cost and it's relatively cheap. And the material, if it's in the gene bank, can then go out to characterization and evaluation. So that's an important, it's taken us years to work that out. And it sounds so simple to explain in a few minutes, but it's with a nightmare. Okay, so then the last thing, and this is the stumbling block. So I am getting near to the end. This is the stumbling block that's happened with crop wild relative conservation and setting up the network is the governance. So we had basic problems setting up the governance structure. So the governance is the way when you set up the network, it's going to be managed. And basically the problem is you need some organization to provide oversight to the network. In other words, they're investing money and they're the ones who are saying whether or not the samples can go into the gene bank as in situ samples, etc. And if you're talking about Europe, then there are various organizations. This ECPGR, which is the European Cooperative Programme for Genetic Resources, the European Environment Agency, and the EEC itself has its own in situ network, which are called the Natura 2000 sites. So we still have a lot of those sites in the UK. So potentially, I thought originally the Natura 2000 sites would be the best bet to do this. But unfortunately, by going for the Natura 2000 sites, it meant that the ECPGR became less enthusiastic about being involved. So there are dense politics involved in sorting this out. And the same is true at the global level, because the obvious organization to set up the network and provide the governance structure at the global level are the Food and Agriculture Organization of the United Nations. Okay, so whatever system of governance you have, ultimately, the material always has to stay under the control of the government. Because in the CBD, it changed the strategy for conservation in that each country owns all of the biodiversity within that country. And you can't just give it to another country. It has to be negotiated, the passage of material from one country to another. So this national sovereignty is a very important aspect. Okay, so this is, I talked earlier saying we wanted to have criteria for whether a site could actually join the site. Obviously, I'm not going to go through all of this, but it just shows that we do have those criteria worked out. We do have the process. We start off with our in situ populations. They're then assessed against these criteria. If they pass that assessment, then they can become part of the network. And then periodically, they have to be reassessed against the criteria. We have various useful tools. And you can see in the top right-hand corner here, we have Farmers Pride. A lot of these have the Farmers Pride logo because particularly within the last EU EC-funded project, Farmers Pride, we produced a lot of end products, if you like. And that's why I was so frustrated in the end. We didn't actually produce the network. But we produced tools for conservation planning, data management. We're able to look up any protected area and tell you which crop wild relatives are present in that protected area in Europe. Management guidelines, evidence-based database on how to manage the sites, etc. All these different information is available. And we also got a significant commitment. So I wanted to show the EC that we did have a strong local commitment for setting up the network. So all of these different spots are various people that have signed up to joining the network. So they manage existing protected areas, or they manage the other types of conservation sites, and maybe some of them are on farm around Europe. You can see there's about 100 here, 100 sites identified, all of various kinds, and they're spread throughout Europe. Right, so we didn't do it in Europe, but we had a parallel project going in Southern Africa. And we did do it in Southern Africa because Southern Africa has the Southern Africa Development Community, which are like our EC, and they stepped up to put their mouth where the money was or whatever, and said they would set up the network. So I'm very pleased to say the first network of this kind set up globally is in Southern Africa and has already been established. So I'm getting near the end, so I just want to tie up with a few conclusions. So Hawkes, Jack Hawkes was here before me, and thank God when I arrived, he was still around, and we had lots of very interesting discussions about genetic resources. Anyway, he was at probably the first conference on in situ conservation of genetic resources. And in his concluding remarks, he said himself that in situ was very much in its infancy. And I think at that stage, he was absolutely right. But since that work, there's been a lot of work done in terms of refining how to do in situ conservation. I really genuinely believe we're at the stage now of implementing the network. And so that's what we're looking for. So we're looking at next steps, just to finish off. So as I said, we've been lucky in having a whole series of EU funded grants and some Darwin Initiative grants. And as I said, the last one I think was the most significant, Farmers' Pride, simply because we were able to finish off a lot of the products and provide a lot of the answers to how you actually do it. So in terms of moving forward, Sheila and I had a discussion with Annette Schnegens, who's a senior policy officer at DG Agri, and we were able to put forward a substantial case for setting up the European network. And so we now are fairly confident that in the next call for the EU funding, there'll be 12 million allocated, sorry, 10 million allocated for 2 projects to actually set up the network. So we've made real progress on that. In terms of, so that's at the European level. At the global level, we're still working with FAO to set up what will be called the Vavilov network, named after Vavilov, as I said, the guy that unfortunately was starved to death after working on food security his entire life. And that's ongoing, the work with FAO, but we are in the short term, we're working with the different CGIR gene banks, and for each one of those, trying to work out the in situ priorities for working on their mandated crops. So there is this global set of gene banks, UN-based gene banks around the world. There are 11 of them. They get a substantial international funding, and they, at the moment, work entirely on ex situ conservation. So I want them to work also on in situ conservation. And so we have a voluntary project we're doing at the moment with them, collaborating to identify which are the in situ locations which are associated with the crops that they work at in the gene banks. And so that, I think, will be a very substantial paper and piece of research when it's done. In terms of the UK, even good news here, in that we've just heard from DEFRA. DEFRA just given us a small grant to fund a PhD, and that PhD will be specifically looking at setting up a crop wild relative conservation network for the UK, which is co-funded by DEFRA, Natural England, and the German Ministry of Agriculture. So let's not speak too loud why the German Ministry of Agriculture is funding this work in the UK, but we won't turn any money away. And also within that context, I got myself elected to the ELMS Committee. So the ELMS Committee are the environmental stewardship scheme set up by DEFRA that pays individuals for doing biodiversity work on their farm, on their land that they own. And part of that is sorting out how you actually pay them for what they do. And so the reason I went on the committee is simply, again, so that I could put forward crop wild relative and land race conservation. So that will become a priority, I hope, in the future for that committee, and in terms of implementing crop wild relative and land race conservation in the UK. So I think I've learned one lesson. When I came to Birmingham originally, probably for the first 10 years, I had virtually minimal grants and minimal, or I'd worked mainly on research, but not on policy. So I think the point I learned very early on was that if you want to actually implement what your grants are telling you in terms of science, you have to work in parallel on the policy side. Otherwise, you're never going to be, I can produce as many reports as I like saying crop wide relatives are wonderful, but unless I can make sure the policy is in place to implement that conservation once it's done, then it just won't happen. So I think that's a very important conclusion I've come to at perhaps in the later parts of my career. Okay, so a postscript. I put this here for Jerry. I know Jerry likes quotes from Darwin. And so Darwin said, "It appears strange to me that so many of our cultivated plants should still be unknown or only doubtfully known in their wild state." Darwin, talking about crop wild relatives, he's puzzled by why no one's working on them. And I'm going to be cheeky and paraphrase Darwin and say, it appears strange to me that when so much is known about wild relatives of cultivated plants, the adaptive genetic diversity they contain, their significant economic value, we have an estimated annual value of crop wild relative use of $115 billion per year. That's not a trivial amount of money. Huge amount of money. So their significant economic value, the threats they are currently facing from genetic erosion extinction, that why, if that is the case, why are we so few resources focused on their active conservation and use, given their overall value at national, regional, and global levels? The time is surely right to invest in in-situ or an integrated national, global, in-situ crop wide relative conservation networks for humankind to sustainably benefit from these natural resources. Okay, I can see that that's the end of the talk. Thank you. But before I go, as is tradition, I wanted to say some thanks for support to various people. Now, usually when it comes to this stage of an inaugural, the inaugural would list A, B, C, and say what they've done. Now, in my case, it's very, very difficult to do that because I honestly have collaborated with thousands of researchers to do this work. Imagine if you're working at a national level, at a European level, and a global level, there are really thousands of them. I'm not exaggerating. So the sort of people I've been collaborating with are present here in these slides. And I would say that there is only one person, I'll point out who that is later on, one person who is in every one of those slides. So who specifically, if am I forced to pick out people, who would I pick out? Well, Vavilov himself, it's a shame in a way we don't have time to talk anecdotes about Vavilov. Very, very interesting character who died a tragic death, but was very much ahead of his time. I'll give you one anecdote. So the revolution happened. After the revolution in 1917, all the Bolsheviks wanted to be seen as common men, so they would all queue up in bread queues to get their own bread rather than have someone do it for them. So it happened that Trotsky was in the queue for the bread in St. Petersburg. And he was not that many people away from Vavilov. So Vavilov at that time was 22, and I don't know how old Trotsky was, but not that much older perhaps. Anyway, he jumped ahead. Vavilov got ahead in the queue, so he's standing next to Trotsky. They were 2 hours in the queue, and during that 2 hours in the queue, queuing up to get the bread, he managed to convince him to give him the money to set up an institute on food security for the Soviet Union. Now, this is 1917. I mean, there must have been huge other priorities that the money was going for. And he got a substantial chunk of money to work on food security. And you might say it was very foresightful of Trotsky, that Trotsky realized that it was going to be an ongoing problem. So all good thoughts for Vavilov. The second is Jack Hawkes. As I said, I came here, Jack was still alive, and I had about 10 years of going to eat at his house once or twice a month and having wonderful discussions. I mean, he was full of stories himself from his own collecting expeditions. but also he was a postdoc with Vavilov, so he knew Vavilov directly. So we had that wonderful link. And it's extremely frustrating to me, for instance, doing that reanalysis of the Vavilov hotspots, that Jack wasn't alive, so I could have discussed exactly how Vavilov created his hotspots, because the text is in Russian. Obviously, I don't speak Russian, but in translation, it's very difficult to work out exactly how he did it. But I think we got there in the end. So Jack, also highly honored. Brian, a good friend, collaborator on many projects. I thought, in a sense, we were perfect partners, perfect research partners, in that he worked on the more genomics elements and I worked on the more field and policy side of things and the genetic resources side. So thank you, Brian, for all of your collaboration and input over the years. My first PhD student, Aishan Dulu, who has carried on since finishing his PhD. He's worked for Biodiversity International and other UN agencies, and we've worked together ever since. And perhaps a lot of the funding I've got for research, certainly international funding, has been because of collaboration with Aishan. So thank you very much, and a very nice person. But actually very difficult for the two of us. The two of us are co-chairs of the Crop Wild Relatives Specialist Group, and Aishan is about this tall and extremely thin. So whenever you have a joint picture of the two chairs of the Crop Wild Relatives Specialist Group, he always feels humiliated. And so he's taken to carrying a sort of something he stands on when we're being taken pictures of side by side. Then Joanna Magos-Briam, someone I've been working with for many years. Again, I hope now becoming her own, a true researcher in her own right. But Joanna and I have collaborated for a very long time, and we've done a significant amount of work, and I owe her an enormous practical debt of gratitude. And then finally, the only person which is on every one of those shots I showed you before is Sheila Kell. So Sheila Kell also, well, again, we're perfect partners in the sense in that Sheila dots the I's and crosses the T's, and I'm much more of a, I try and, I'm all over the place, perhaps disorganized in some ways, but I do love the subject and going forward, and Sheila's the one who points me in the right direction and gets me going. So thank you, Sheila, for that. I really appreciate that. Right, so I think that's that. Oh, and the very last slide. So this was just to finish up and say that the people I haven't thanked are the farmers. I spent, before I came to Birmingham, I spent 10 years working as a collector for FAO. So I spent time traveling around the world, collecting plants, meeting farmers, working with farmers, and I really enjoyed that. And also on the in situ side, if you're doing on-farm conservation, then again, you're working with farmers, talking to them, collaborating with them, having tea, endless tea, or drinks. And then if you come out of one house, you have to go into the next house. You know, it's an amazing situation. But we couldn't do the work on genetic resources. It's for their benefit, but it's also enormous joy to be able to do this. So thank you very much.
So thank you, Nigel, and congratulations.
Thank you.
So my name's Neil Hotchin. I'm the outgoing head of school, and I think it falls to me to just say a few words at the end of your inaugural. So I found that a really excellent and informative talk, but in some ways very sobering. Certainly the beginning of what you were talking about really had me thinking, although I think you gave us grounds for optimism.
Yes. Towards
the end. I mean, you talked really convincingly about the need for policy intervention, and your point at the end about it's okay doing experiments, but really you need to influence people. There needs to be impact, and so on was really well made. Working on establishing networks, I mean, that you've glossed over that quite quickly, I think, but that's an amazing amount of work over a large amount of time, I've no doubt, and so on. Talked about very convincingly about the need for in situ conservation. I guess I'd naively thought we could just bung all the seeds in a box in Norway and everything would be okay and so on. But I think you very convincingly made the case that's not the point and so on, or not the solution. I liked your stumbling block being governance as ever. But then you ended up talking about reasons to be cheerful in a way, I suppose, that yes, progress is being made. South Africa being a really nice example of how it can work, and hopefully it can move on from that. So that was, because I didn't really know what I was doing here, I also made a couple of notes as well. And I was thinking that as researchers, I guess we all deep down hope that something we do or discover will at some point have an impact, however small. I don't think anyone can have listened to your talk or followed your work and not thought that what you're doing is going to have major impact. I think generations to come, there'll be people exceedingly grateful for the work you and your colleagues such as Sheila have done and will continue to do. I mean, that was a really nice thing that you're going to carry on doing this as well. I think I'll finish there, other than to say that I'm really pleased that we're able to start these inaugural lectures again, that you're the first one after the hiatus, and an important part of school life, a way of celebrating the work we do, celebrating our professors. And on that note, if people want to make their way to the Lapworth Museum, then there will be some drinks and hopefully some food, and you can talk to Nigel in person and ask him more. Thank you.
Thank you. Thanks, Neil.